BCS webinar on this last day of June, which also marks our entry into the second half of 2020. So hopefully everyone is doing well during phase 2 while we're still working from home or have started returning to our respective workplaces. And of course, at BCS, we're still embracing virtual events, and we're very glad to see that there are lots of people still participating actively in these online forums. So just a very quick intro for those of you who are not very familiar with BCS, Biotech Connection Singapore. We are a non-profit organization, and our mission is really to promote and support life sciences entrepreneurship. So besides events, we also have a consulting services arm and a communications arm. So if you're interested, just check us up on our website or sign up to our mailing list, which is right on our homepage, or follow us on LinkedIn. So today will be quite an interesting and packed event. So let's get to it straight away. So just want to start off by saying that this event is co-organized with Twist Bioscience with additional support by Merck. So we're very thankful to both companies for supporting us. And we put this together because, as we all know, the COVID-19 pandemic has resulted in global and local efforts from both academic and industry research sectors to study the biology of the virus, of the disease, and the underlying immune responses that are caused by the infection. So in this webinar, we would like to touch on the ongoing efforts in developing both therapeutic and research solutions for COVID-19. So I'll start off with just introducing our speakers first, before passing some time over to Merck and Twist Bioscience to introduce their companies and their product pipelines before we go straight into our panel discussion. And just a quick housekeeping, as you might see, the attendees will see that the chat is disabled. So if you have questions, please type your questions in the Q&A tab. You can do that anytime, and we will hopefully address them in the last 20 to 30 minutes of this webinar. Right, so to start off with, I would like to welcome Dr. Eong, who is a professor and deputy director of the Emerging Infectious Diseases program at Duke-NUS Medical School. So Prof. Eong plays a key role in ongoing efforts by Duke-NUS to develop a vaccine for SARS-CoV-2, the COVID-19 coronavirus. So he's instrumental in various ways, for example, through the partnership that was signed between Duke-NUS and the US biotech firm Arcturus Therapeutics that will see the development of a COVID-19 vaccine. And locally, he's also co-founder of TICAN, a local biotech who is at the forefront of conducting phase 1 clinical trials for a potential treatment for COVID-19. Next, we are also very honored to have Rajeev Nair, who is currently Senior VP and Head of APAC Research Solutions for Life Science at Merck. And he has over 30 years of sales, leadership, and general management experience in the life sciences industry. So he has over 20 years with Merck and Millipore, and he has held leadership positions in various businesses within Merck Millipore, as well as worked in several countries across different continents in Asia, Europe, US, and Latin America. So thanks, Rajeev, for joining us. And finally, we have Mark Dupal, who is a Senior Sales Director for APAC at Twist Bioscience. And he's responsible for the sales organization and driving the brand awareness and growth of the company throughout APAC region. He has over 20 years of expertise developing and commercializing consumables, capital equipment, services for the life science and diagnostic industries. And he has a strong focus previously in NGS, imaging, and precision medicine. So again, thanks, Mark, for joining us. So right now, I will pass the time over to Rajeev, representing Merck, to share a bit about your company and your technology pipeline. So BCS, we will share the slides. Right, you see it on screen now. So Rajeev, please.
Yes, thank you, Natasha, and good afternoon to all of you. I'd quickly like to just take you to give you an understanding of what we do and also what we do with regards to COVID. So Merck is a global science and technology company which was founded in 1668, about 350 plus years ago. And today we have 3 distinct businesses: healthcare, life science, and performance materials. Within healthcare, we handle prescription drugs for multiple disease areas, but primarily in the oncology space, we have products for immuno-oncology, oncology, like Bavencio or Erbitux, which is for colorectal or head and neck cancer. We also have for multiple sclerosis, we launched a new molecule called Mavenclad, which helps the patients because it's a tablet that you take for 2 weeks for 2 years. And this tremendously helps the patient care. Then we have what's called the life science business, which I'm part of. Here, our role is to support the scientific community to empower breakthroughs in science by making research simpler, more productive, faster, and more exact. And this means we have a range of innovative tools. Some of you might have heard of Immobilon Go, which is a new small device, plastic device that's used where you just put the Immobilon membrane, you put in your primary and secondary antibodies, leave it, and the next day you come, you can actually develop and see the bands. There are also innovative devices like Scepter, where you use as a cell counter, very convenient for the scientists. We have services. When I say services, it's in the biomanufacturing development area for monoclonal antibodies or vaccines. And we have these labs also in Singapore called the M Lab, which is available out here in the Science Park. Then we have digital platforms. When we say digital platforms, one is, of course, easy to procure e-commerce platform. We have also content where we have close to 400 million page reviews every year. So we have reasonably a good scientific content up there, and we enable scientists for easy procurement. We have retrosynthesis design software for designing your chemical molecules. We also have single-use platforms which make flexible manufacturing much more simpler for MABs and also in the vaccine space. And finally, we have performance materials where we have very innovative solutions for liquid crystals and OLEDs. But what's even more important today is in the semiconductor solutions, where we provide a whole range of chemicals and also work with the Apples and the Samsungs of the world when they design the next gen equipment, IoT equipment, and we play a role in the chemical design. And we have specialty chemicals. Next slide, please, Natasha. So when we look at the COVID-19 and how do we play there, all three business sectors I spoke about, we play a role. In healthcare, for instance, we just are going into phase 2 with a molecule, the TLR7 and 8 inhibitor. Primarily, this is to reduce the inflammation response before you have the cytokine storms. So this is going into phase 2, and we hope this comes out positive. In the performance materials area, before I go into life science, I'd also like to share, we are a critical component for the wafer fabs, and hence we were running and enhancing our production because with everyone working from home, there is more demand for IoT devices, etc., and we played a role. Coming to life science specifically, we are in the virus detection area where we supply critical components for viral transfer medium, viral extraction, detection kits, etc. In the virus characterization area, we provide a range of biological reagents and hardware for virus basic research, for cytokine storm, for applications like Milliplex and ELISA kits. We also have hardware on which you can run all these kits. And finally, when you look at vaccine and therapeutics, most of you know there are close to about 7 different technologies that are emerging in the vaccine space for COVID-19. And within that, we supply nucleotides, capping enzymes, RNase inhibitors, etc., and also a range of products that go for the therapeutics. So overall, we claim, and I would say in the last 30 years, every drug that's possibly passed through US FDA would have touched a Merck product somewhere in its lifetime. With that, I hand it back to Natasha. Thank you.
Thank you very much, Rajeev. Right, so I'm sure we'll ask more questions. about what you're doing at Merck shortly. So now I'll pass it on to Mark from TWIS Bioscience.
Thank you, Natasha. Thanks for allowing me on the call. So next slide, please. So not quite as old as Merck from 1668, where TWIS was founded in 2013 with the advent of our new oligo manufacturing capability. And we have a number of sites throughout the world. San Francisco is our headquarters. In APAC, we are headquartered out of Singapore, but we also have sites within Tokyo, Japan, and we also have a site in Shanghai. In 2018, we went public and we now trade as a public company. So next slide, please. So TWIS is really based on some novel technology that they've created around oligosynthesis. That's really the basis of the company. If we look at the left-hand side, this would be considered traditional oligo manufacture, where we have one oligonucleotide per well in a 96 well or 3 out 4 well plate. Where TWIS have really revolutionized the technology is using a silicon wafer platform, no bigger than an iPhone really, where on that platform, using microfluidic devices, we can print up to 1 million oligos with incredibly high accuracy, you know, with about 1 in 3,000 base pairs errors being introduced. So you can see the scale where we can now massively parallel oligosynthesis, which helps us within all of our business verticals. Next slide, please, Natasha. So out of the technology is born 5 different business units. We have a genes or clonal genes business. We have oligo pools. We have libraries which allow people to, or researchers, to engineer proteins. We also have an NGS section where we use target enrichment to pull down specific areas of the genome for NGS sequencing using either Illumina or NGI technology. And kind of the big play at the end, where Rajeev and I were talking previously around our data storage technique, where, you know, DNA is a great medium for very long-term storage, and we have a number of very large collaborations going over in the US where we're using our high throughput oligo manufacturer for data storage of, you know, pictures, movies, etc., you name it. Next slide. And finally, for the tools that we use to address, you know, the current epidemic, we use 3 of our business verticals. We obviously have the genes or the genes and clonal genes business to produce synthetic RNA controls that can be used as positive controls. We have NGS libraries, target enrichment panels for research panels for SARS-CoV-2, and also coming out with a new respiratory panel. And then finally, we have TWIS Biopharma, which is like a contract research organization within TWIS that allows us to produce very, very rapidly a number of therapeutic antibodies, which we have designed against the spike protein and the ACE2 receptor. So that's probably enough at the moment, but thanks, Natasha.
Great. Thank you very much. So thank you both for sharing with some slides with us to give a very good overview. So right, let's get straight into the discussion. So I'd like to start off with Eong first. Yeah, there's lots to talk about. So maybe to start off with, I'd like to ask you, so since the start of the outbreak, which, you know, in December 2019, how have you seen your research focus change? And can you share about some of your key efforts in tackling this coronavirus pandemic? And perhaps you can comment both in terms of from the perspective of your research lab at Duke-NUS, as well as your involvement in TICAN.
So first of all, thanks very much for having me on this panel. Pleasure to be amongst very distinguished colleagues. So I actually work mostly on dengue, and my primary research is on dengue and related flaviviruses like Zika and yellow fever. And how we got into this vaccine field is really by chance. I mean, for a long time, the problem with dengue is that there's no animal model that really replicates what happens in humans, right? And so studying dengue then becomes very difficult because you can either use in vitro systems or you rely on animal models that don't really tell you what happens in humans. And if you do recruit dengue patients into the study, then between infection and the point where they develop fever, about 5 to 7 days would have passed. And in that 5 to 7 days, a lot of things can happen. And that could shape why some people get disease and some people don't. And so, we're not able to understand those processes. Then by chance, we stumbled on the use of yellow fever yellow fever vaccine as a way of understanding what happens to dengue patients. So we've been using yellow fever vaccine in human volunteers as a way of looking at why do some people get fever, a bit of body ache, a bit of headache after they get yellow fever vaccine, whereas the other half of the people who get vaccinated don't develop any symptoms at all, and everybody gets immunity. Right? So what gives rise to this side effects? But the side effects look very much like a flu-like illness, which is like the start of dengue. And so that got us into this area where we now can begin to understand why some people get sick and some people don't when they're exposed to the same virus. And as a cause of that, we also got to understand vaccine. Why does it work and what causes the side effects? How do we look at vaccine safety from a genetic point of view rather than from whether patients report these symptoms. So that was where we at until end of 2019. And so then when COVID-19 arrived in the shores of Singapore and looked like it's going to be a bigger problem than SARS in 2003. So naturally we had to come up with solutions, and instead of working in the field of diagnostics and all that, we thought the real gap and what would really solve the problem would be either a vaccine or a drug or both. That would prevent infection, and if you do get disease, then at least prevent you from getting severe COVID-19. So that's how we got into this, because of what we have done with dengue and yellow fever vaccine and all that. We're now using or bringing all that we've learned into play to tackle COVID-19.
Right. So what about the activities and that your biotech company TaiCan is doing, and how has that changed? Based on before this whole thing happened.
Yeah. So TaiCan was formed largely to tackle this idea of epidemic, a response to epidemic. And really the crunch, what we felt that the real bottleneck in any therapeutic development is the long time between the point where you have a candidate to the point where you get into humans. And that's usually, before we got into this game, as far as I understood it, it ranges from somewhere between 3 to 5 years, and that's very optimistic. It could take longer than that. So that kind of timeline isn't appropriate for epidemics. I mean, certainly when you compare to SARS was in Singapore for all of 2 months and then it was over. It never, So if you really want to bring a therapeutic to tackle a vaccine, it cannot take 3 to 5 years. It must take several months, right? So that was how TaiCan came about, to see, can you tackle this in a way that is more relevant as a response to outbreak. So the first one we took on was Ebola, because of the West Africa outbreak in Ebola, and there was a risk that travelers from West Africa could bring the virus into Singapore. And there we learned all the problems with therapeutic. That's why you never heard of TaiCan developing Ebola therapeutic, because it was just hurdle after hurdle after hurdle and roadblocks here and there. So then we decided that we will do another one, and everything had to be in the context of an epidemic, because otherwise the regulators will not say that there is any urgency, right? So it had to be an epidemic. So the next epidemic was Zika. And so that was an opportunity for us. And fortunately, because Zika is related to dengue, we kind of knew where the target should be, learning from dengue, right? So for Zika, we managed to shrink what would take 3 years to 15 months to get from a candidate into humans in 15 months. And then the next outbreak wasn't in Singapore, was in Brazil and Africa, was yellow fever, and again related to Zika, related to dengue. And so we could take what we have learned with Zika and now bring it to yellow fever, and we did it from point of candidate to human trials in 7 months. And for yellow fever, we actually finished a trial in 1 year, in 12 months. And now with COVID, again, it's this iterative process that the more we do, the more we understand what's really necessary and what's really not needed, because it doesn't inform us in any way about what's going to happen in humans when you put this drug into human volunteers. So again, with that data, we could go to the Health Sciences Authority, our regulators, and say, perhaps this is where we should focus on. This is the kind of safety profile. Again, we brought in genetic tools and all that to look at purity of the cell lines, the purity of the deep analytics to look at the purity of the product, look at toxicology in animals, and now in human phase 1 trials, also look at how safe this thing is. And so we're able to then again take all that we've learned and build on that. And so we went from candidate to clinical trial in 4 months, in 3.5 to 4 months. So, but if you ask me, that's still a few weeks too long. I mean, in 3 months, I mean, how many people in Singapore had COVID-19 and how many people died from it? So I think we can still do better. And the idea is that to build up that data that will then inform us on what needs to be done, what's critical to inform us about whether this drug is going to be safe and whether it's has a potential of working in humans, and then use data to drop the rest that really doesn't tell us anything about what's going to happen in humans.
Mm-hmm. So yeah, I mean, it's a very exciting time. So as you mentioned just now, that during the SARS outbreak, more than a decade ago, that the outbreak came and went in just a couple of months, and most definitely before a vaccine can be made. So with regards to COVID-19 now, how do you think the situation will compare? And the million-dollar question behind everybody's mind: when do you think we'll expect a vaccine or a new therapeutic launch in the market?
Yeah. No, I think COVID-19 is very different from SARS. I mean, I think this is here to stay. I suspect that if we have this webinar again next year, we'll say that this is now part of our seasonal flu, perhaps a more severe one. I don't think we can get rid of it. I think it's just too many people have it now. And very unlike SARS, the one thing that was really remarkable about SARS is that you could not find asymptomatically infected individuals. Everyone who had infection had symptoms. And the added bonus was that even when you have symptoms, you weren't infectious until the 2nd week of illness. So the disadvantage was that it was very hard to diagnose SARS because you couldn't find the virus in the 1st week. It only appears in the 2nd week. The good news is that by the 2nd week, most of them were already so sick that they are already in the hospital. So to control SARS from spreading, you just have to make sure there's tight infection control in the hospital. And that's what killed the virus. Right? Mm-hmm. Whereas SARS-CoV-2 is so different. I mean, there's a lot of asymptomatically infected individuals. There's also a lot of people with very mild infection. And so then the community, and the virus is shed very early on. And so then the community spread is so much more prevalent than SARS. So it's a different beast entirely. And I think this is here to stay.
Yeah, so I mean, I worry about asking a controversial question, but are you thinking of something that will come out, not just from yourself, but also global efforts in a few months, in half a year, in a year's time?
About a vaccine?
Yeah, what are we looking at? What do you think?
I think the one thing that we have to say about vaccine is that, looking at what Moderna has done, looking at what the Jenner Institute, the Oxford group has done with how fast they have brought a vaccine into clinical trial, it is very remarkable, right? I mean, never I would not have imagined, despite what we've done with the antibody, I would not have imagined that we could move so fast with the vaccine. So our own vaccine collaboration with Arcturus Therapeutics is going to hopefully enter clinical trial around beginning of August. So it's slightly behind because it's a little bit more complex. Hopefully the complexity will be good. But nonetheless, despite all this speed in getting into first-in-human trials, the difficulty is going to be efficacy. And that's where the complexity comes in. Because I think it's not difficult for vaccines to elicit a short-term protection. I don't think that's difficult. The real hurdle is long-term. How long can this immunity last for? And, and how long will it actually last, right? So when do you need to boost if it's not going to be, if it's not drop below the protective level? That's the unknown. And so if you ask me, when will we know that a vaccine could work? It could be as early as beginning of next year. I don't think we'll make end of this year, but I think beginning of next first quarter perhaps. But then again, will it become available to the public? Again, that's a whole new challenge, because there's going to be manufacturing involved. There's going to be the fill and finish, the pricing, and all that. How we're going to distribute the cold chain, the logistics. All that will each one of those will present some level of delay. But the real crunch is, okay, you roll a vaccine out. How long are you going to protect the population? When it wanes, will you create a problem? Right? Just like the vaccine. In some population, this immunity is not high enough, then that's going to create a problem when they do get infection. So along with vaccination, there has to be a solid surveillance program. The public health authorities must be prepared to invest in a surveillance program so that we catch any problems early and know when to boost the population. So it's not, I think having a vaccine is nice, but it's not the end of the game. There's still quite a long way to go.
Yeah, okay. Thank you very much for giving such a wholesome view of this situation. And with that, I will move on first to the other speakers. So we have a few questions, on the development of, research solutions. So this could be, questions that are targeted both at Mark as well as Rajeev. Is it possible if you could share a little bit about your unique approach in contributing to COVID-19 research? And I know you've covered some of your technology pipelines earlier, but importantly, how did your company decide on the solutions to offer? And are you engaging with your customers to make sure that the products stay relevant and address their needs?
So if I may take this question, when the pandemic hit all of us, the first thing was for us to first understand what's happening around. And for this, what we did was to create a task force first to interact with our customers to understand where is it that we could play or support them. Two, within the company, we got together the technologists. We also got data scientists to just understand how the disease has been spreading around and arrive at what are the key problem areas that we as a company, looking at the technologies that we have close to 300,000 products that go into the research area. And so hence we had to get our arms around what the problem is, where could we support? And finally, we came to the conclusion that there are three areas that we would like to focus on. One, the diagnostic space, another is therapeutics, and third, vaccines. And then what do we do in these three areas? So for example, in the diagnostic space, we supply a whole range of consumables that are used by diagnostic kit manufacturers. We also supply, for instance, I was telling, viral transfer medium. So when the swab samples are taken, it needs to be carried for testing. So for that, there are components that go into that, which is the HBSS solution, there's FBS, that's fetal bovine serum, or gentamicin. So we supply these products. Then the virus extraction buffers. There are components there like guanidine thiocyanate, twin 20, etc. So for each parts of the workflow in the diagnostic space, we identified what could we support. Once we understood what we could do, the demand was so huge that we had to be fair to our customers around the world. So that called for putting together a team that clearly worked on allocations so that we are fair to our customers globally, because this hit the globe almost at the same time. And the third thing we had to do was we couldn't meet the demand, so we had to repurpose some of our production lines so that we could enhance the production, which took us some time. That was in the diagnostic space. Similarly, we also looked at for the basic research area or for the clinical space where they are trying to understand the cytokine storms. Here we had milliplex kits, etc., that could be used. So ramping up production in that area. And finally, when we come to the vaccine space, through the process solution business of ours, we do supply pilot scale all the way to fill and finish, complete manufacturing solution. But the advantage that we provide is we can manufacture flexible single-use systems. So the traditional manufacturing was stainless steel hardware, process line. Now in a pandemic like this, where there are many countries who want to start the manufacturing vaccine locally, it becomes important to have a flexible solution. And that's what we're focused on, to manufacture those flexible single-use systems, pack it into the boxes, and they could be shipped anywhere around the world. And through this, we think in all these three spaces, we've been in a position to provide some value to our customers, which help them in their development.
Thank you. How about Mark?
Yeah, thanks, Natasha. I guess, you know, Twist's approach was to look at what can we do immediately to help, I guess, our customers, but really, you know, this was a global engagement immediately, but also in the future. So one of the things that immediately came up was the requirement for synthetic controls. You know, it's dangerous, obviously, taking live virus, extracting the RNA and using it as a positive control within a number of different diagnostic tests. So we took it upon ourselves to take the current SARS-CoV-2 genome, synthesize it, and we were able to do that, creating 6 overlapping fragments. And we provide that, you know, as a positive control. And that was one of the very first things that came out of our kind of task force. Now we have 6 available, depending on how this thing is emerging and changing, with a 7th coming on board. We also really looked at what do we need to do in the future? What's humanity actually going to need to try to combat this? And you know, Prof mentioned about surveillance. So, you know, we came up with an NGS target enrichment based solution for the surveillance of the different viruses, or sorry, the SARS-CoV-2 virus within the population. And now we're coming out with another one, which is a more respiratory virus panel, which allows us to look at not only SARS-CoV-2, but other respiratory viruses, up to 29 different viruses, which will allow us and I guess researchers to understand, you know, if you've got a co-infection, what happens to the patient. And then finally, you know, we work as a kind of like a semi-CRO, where we're able to work with a number of customers at Proteona, or a small startup within Singapore, on identifying neutralizing antibodies from patients who have been infected. And we can very, very rapidly produce IgGs at very high quantity and large numbers, which allows us and researchers to actually understand, can we produce a neutralizing antibody before we get a vaccine in place?
Mm-hmm. Right, so I think both of you touched on a point that I want to sort of discuss further, which is, you know, working in this very fast-paced environment. So, I mean, in terms of the needs in the research community, what are the greatest needs that you see going forward from now, and what strategies have your companies put in place to try and be very quick with, you know, releasing products, developing products in this, you know, very fast-moving space? And as well, that also applies to, you know, designing tools to study this virus, because we are aware of, you know, the different variants, mutations. So this relates to surveillance as well. So how together are you trying to tackle this changing environment?
Rajeev.
You want to take it, Mark, or should I?
Look, I can quickly provide some answers. So we're working with a number of you know, laboratories globally, you know, Cornell Medical School, the Broad Institute, and you know, this is an ever-evolving situation that's happening globally. So, for example, the respiratory virus panel that we have coming out, that was in request really from a lot of the customers that we have, and it really is, it's a question out there, you know, what happens to patients when they have maybe influenza A and SARS-CoV-2? What's the outcome? I mean, that's an important question that needs to be studied and understood, because if we can understand that a patient does have a co-infection and we've studied it and we know how to treat them, then we have a much better chance of saving patients who may have a life-threatening, you know, life-threatening infection.
Thank you.
So within Merck, the approach within the life science business that we took was first and foremost to look at the three pillars that we are talking about and identify core groups that would start working with these accounts. So let's say in the case of therapeutics, so two areas, that's the antivirals or monoclonals. So that was the space where the research team would work with scientists in the laboratory and identify which part of the workflow that we could support them with. At the same time, we had colleagues from a business of ours called the Process Solutions business that's working with most of the vaccine manufacturers, like for instance, the Jenner Institute, etc., where we are looking at how we could help them in their flexible manufacturing, scaling up the vaccine production. So that was the approach that we took. And this, I feel, has helped our customers in a big way to also ramp up whatever research activity they were doing, and also in the manufacturing space.
Right. So I guess now, I mean, I'm interested to hear more from Merck from an MNC perspective. So you did mention a little bit just now in terms of you know, your very global efforts, and you have a wide range of solutions in the life science space. So what is I mean, can you comment on whether there are different needs in the APAC region from what you're seeing abroad, such as in Europe, or are there sort of synergies, you know, in terms of how Merck's strategy in terms of developing products for needs globally as well as locally? How are you sort of segregating that or combining that?
Okay. So when we look at the landscape of our services and manufacturing globally, we have close to 67 manufacturing plants around the world. And when we look at now the Asian continent, and here is where we see a lot of emerging opportunities, particularly when you look at countries like China, where are not only the regular monoclonal antibodies, but they're talking about novel therapeutics that they're working on. And that's an area of interest for us in the cell signaling area and cell therapy area. Looking at Singapore per se. So here we started working closely with EDB in 2005 when the Singapore government was deciding on creating a hub for biomedical in Singapore. And what we understood at that stage is that we could partner with the government to equip a training institute, to equip Singaporean professionals to move into the biology space. That would be an interesting area. And with that, we set up what's called the M-Lab, where we train professionals for downstream processing. And that's in the area of clarification, purification, validation, etc. And till date, I would say we have trained close to 1,200 professionals in that space. How did it help? It helped Singapore because we had Roche, Lonza, Amgen, Novartis, many of the large multinationals who moved in here for their manufacturing base. Anyone joining the company used to come to our lab to get trained. So that was, I would say, a big effort that supported the emergence of bulk manufacturing in Singapore. Although one of the challenges that still remain is fill and finish doesn't happen here. The bulk is shipped out to Europe and US where it's filled and finished. Another lab that we recently opened with an investment of close to 8 million euro is our bio-reliance lab. Here, what we do is we do lot release testing. That we conduct the biosafety testing for 3 parts of the process flow. One is the in-process bulk harvest. When you have the bulk that you manufacture, you need to test it at various levels. So we provide these services within our lab in Singapore. Second is for the drug substance. So you have the active ingredient which has been purified, and that needs to be tested to ensure that there are no toxins, bacteria, viruses, mycoplasma, etc. And finally, the drug product that is there, we also need to ensure that it meets the biosafety standards. So with these capabilities that we have built in Singapore, we have similar capabilities in China. We also have an M lab in India and also in other, for instance, in Korea. And this is helping the local pharmaceutical industry, biotech industry, in equipping themselves in developing their molecules.
Right. So just another additional point. So as as a global life science supplier, you've mentioned multiple countries. Did you face any sort of supply chain issues? I mean, it's probably going to be yes, in rolling out these solutions across the globe, especially, since a couple of months ago. And how did that impact your business, and what measures were there to mitigate these issues?
So definitely the answer is yes. There were major issues, not just within our manufacturing, but also in our sourcing. Because one is to source the components to manufacture the product and then ship it around the globe. So this is a lesson for us where we now need to revisit our supply footprint, our supply chain footprint. That was the point number one. Point two was capacities. When you're faced with such a pandemic, there is a surge in demand where customers genuinely want products, and we do not have the capacity available. But here I would give a lot of credit to our team within the supply chain and manufacturing, where they immediately identified process flows, and they repurposed some of the manufacturing lines so that they can enhance production of components or products required for COVID-19. And that was something remarkable, which again, we have never done in the past, and this is amazing. Now I would say the situation is far improved, and I would say going forward, the supply logistic channels have opened up, our manufacturing capacity has opened up, and I'm sure customers will get better supplies.
Right. Thank you very much, Rajeev. So back to Mark from Twist Bio. So, I mean, some of the questions apply as well. So I'm interested to know, as a company which is initially based in the US, and now you have a footprint in Singapore, and you're covering the APAC region, are there differences in the needs that you're seeing? So that's the first question. And the other question is also, I'm interested to hear about your perspectives on supply chain issues around the region and globally that your company face.
With respect to differences within the regions, the only difference that I guess I can comment on is when the infection actually broke out. So obviously this began early January in China, and we started working very closely with the CDC at that stage in implementing, not only our controls, but our NGS target enrichment panels to actually, understand the genotypic nature of this virus. So we're seeing a steady uptick, unfortunately, as the virus started to take hold throughout the region and globally. We could just see that, the spread of product really tracked with the spread of the virus, which is an unfortunate thing, but that's really what happened. So, yeah, I can't say that there was much difference in terms of the demand globally, other than just when it occurred. Yeah. With respect to how Twist has dealt with, the epidemic and being able to continue manufacturing, we were lucky enough being based in San Francisco that we would consider it an essential service, so which allowed us to remain open. And really this comes down to the resilience of human beings, to be honest. If you're faced with an unknown virus, to be honest, and you're asked to go to work, it's a difficult task, right? So we did what many other companies did. We split shifts, we stayed home if we needed to stay home, but the resilience and I guess the professionalism of the manufacturing and the R&D group, they really needed to be on site and be there continuously to be able to not only develop products, but make sure that we could supply COVID-related products, but all of the other products that we still supply to our customers, in oncology testing, etc. So, touch wood, we were lucky enough at this stage to remain open. The business is still doing well. And again, as I say, it's kudos to the people in manufacturing and R&D that are really kind of holding the flame alive at the moment.
Yeah, right. So I guess we're also interested to know, I think you mentioned Proteona just now. So I'd like to ask, from Twist Bioscience, what's your unique role in this international alliance that's led by Proteona in developing neutralizing antibody therapy? And how did this collaboration come about? And how do you foresee, the different members of the alliance working together?
Yeah, it was kind of serendipitous, to be honest. I remember waking up one Saturday morning and seeing their name. So really, I sent an email out to the CEO, Andreas, and he responded, and we started having a chat about, hey, look, where can we help? And these things happen, right? In time of crisis, you get on the phone, you get on a Zoom call, and you start talking to people to see how we can help. So, we really needed to understand what was happening between, 10x Genomics, Novogene, Proteona at that stage, and where Twist could come in and really add benefit. I think first and foremost, the goal was to really assist immunocompromised patients with some form of protection against SARS-CoV-2, because the last thing you want is to be dealing with, a cancer and then going into a hospital and having the possibility of picking up an infection. So the goal had to be clear. And I think we came up with that strategy quite quickly. The Proteona are experts in single cell biology. They really are. They can understand what's going on, from a transcript level, which then translates into a protein level. So working with obviously 10x Genomics in the isolation of the single cells and the transcripts, Novogene providing the sequencing platforms and the service. And then where we came in, where we thought we could add value, is taking these novel transcripts from humans who had recovered from an infection and then producing the IgGs very, very quickly. So we're at a stage now where we have our first candidates coming through. We're starting to manufacture IgGs, which will then go back to Proteona for some downstream analysis with some of their other collaborators in the oncology units of hospitals within Europe.
Right, thank you. I mean, it's great to hear, all these, partnerships going on. And I feel very fortunate to be in the, biotech industry myself, and even in Singapore, essential services. And I'm really hoping to see, some good news coming out of the different collaborations. So right now, I mean, I have a couple of questions that could be addressed to all three speakers before we move on to the live Q&A. And one of them is that, there is a lot of news about potential breakthroughs in research, new clinical trial candidates, and thousands of papers and findings being reviewed every week. So this pace, of research and publication is very different, of course, from how, we normally conduct research. So as scientists, or, as developers, what challenges do you face in maneuvering through such a research landscape, and what steps are you taking to keep up pace without sacrificing research quality and scientific rigor? And I mean, in terms of the industry, the kind of products that you put out there that are, backed by solid science. So I think any of you, Eong, Mark, Rajeev can step in to answer this question.
I'll let Prof.
Okay, thank you. No, I think the rapid release of manuscripts and all that on bioarchives and other online platforms have been good and bad. Good in that, you know, the information is not hidden and until much later, and it's made more available more rapidly. But the bad thing is, of course, a lot of them are not peer reviewed, and you know, trying to sieve it through the signal from the noise it's been really challenging. So whilst there's a lot of very interesting findings out there, there's also a lot of junk out there, and navigating it has been is tough. So I mean, I guess at the end of the day, it's still nice to read a peer-reviewed paper. I mean, even then, it's not foolproof. But yeah, you got to navigate the bioarchives with a lot of care. There's a lot of misinformation out there.
Rajeev or Mark, any comments? Yes.
Yeah, as a company, this is the time to rely on the purpose and communicate this to the team, which is spread all around the globe, because it's a crisis everywhere. There are emergencies everywhere. How do we as a company with close to about 22,000 employees within our division address this? So first was reinstating our purpose. Our purpose is to support the scientific community in their research. We are a life science tools provider. So understanding that this is an opportunity to live up to their purpose. The second most important thing is to lay down some basic principles so that everyone across the organization understands we are aligned and what the priorities are. First, it's the safety of and health of the employees. No compromise on that. So we know, like Mark was saying, we still have to have our manufacturing R&D folks working day and night, and hats off to them for supporting. But at the same time, the others who are in touch with customers, they need to be in touch, but remotely. So health and safety, both for us and to ensure that we respect the customers. Second is, I've been with Merck for 25 years, and I can proudly say we haven't seen the kind of collaboration that's happened at this moment within the company and with customers, although we cannot meet each other face to face. So this was fascinating. And we could see people also ensuring that living by the principles of integrity and risk-taking, moving ahead when customers need something and ready to experiment to support them. And this collaboration, I think if we can even retain 10% we had during these months, we would be a far, far better company than we are today.
I think everyone can say the same in terms of that. Mark, do you have any comment on that?
The only thing I would say is that, you know, in times like this, you really need to work with experts like, you know, Professor here. You can only be, you know, an expert in so many areas, and in a company like Twist with, you know, 600 employees, we're pretty small, but we're nimble. But we rely on real expertise to help us understand, hey, what are the real needs right now? And what are the needs going to be in the future? And then we can get our R&D scientists to really understand and you know, come up with innovative ideas that really do help in understanding now and into the future how we can help.
Right, thank you. Right, I mean, I'd like to move on to the live Q&A because there are a couple of questions. So let's start. So there's a question directed at Eong, and that is, are there any biomarker profiles that are able to distinguish between COVID-19 or SARS-CoV-2 virus infection with various other viral infections?
If you mean like cytokines and all that, the answer is no. It's not a foolproof way. I think if you look at it from even if you use whole genome kind of microarray to kind of ask whether there are signatures, host response signatures that's very much associated with COVID-19 versus say other, you know, respiratory infections like influenza, you probably can find it. The problem is that it is so dynamic that it is almost impossible to apply it. By that I mean, when we looked at our COVID-19 patients, the first few in Singapore, and just use interrogate 500 genes every day, every single day over the course of illness from the point where they were admitted to the point where they got severe disease, and then after that when they recovered. Every day is different. So when you do get a patient, you have to be very clear what day of infection this is in order to ask, okay, what genes should I be looking at, right? Practically, that's going to be so difficult that it's impossible to implement in a healthcare setting.
Right. Another question for you as well. So the SARS-CoV-2 virus seems to mutate. And does that mean a vaccine or treatment has to evolve according to that? And what are some of the challenges in trying to tackle that?
I think for a therapeutic antibody, that's certainly a concern, especially if the antibody targets a part where the virus does drift. And that's why it's important to know where the antibody binds to in order to know whether there's risk in whether if the virus mutates, whether it becomes ineffective, or more importantly, whether the antibody is going to drive virus evolution. So that's a concern. From a vaccine perspective, I don't think it's that much. It's less of a. I think it's less of a problem. The reason is because it's a polyclonal response, and at least looking at the phylogenetic tree of coronavirus, it doesn't drift as much as influenza. So I don't think it's going to escape the way influenza does, and that's why you need updates in your vaccine composition every so often. I don't think it's going to happen to coronavirus.
Right. Okay. Thank you. Last question, or not last, but another question for you. Do you think there is an underlying host genetic predisposition between symptomatic and asymptomatic patients?
Short answer is yes, because if you look at the age, the disease, the infection in different age groups, the outcome is different. Those with disease susceptibility factors are not comorbidities like diabetes and all that. There seems to be some trends there. Perhaps it's not so much a genetic susceptibility factor as much as an epigenetic susceptibility factor. So I think it's probably influenced by a number of things and not just whether you have this gene or not.
Mm-hmm. Right, thank you. So there's another interesting question that I guess all three panelists can share about. So what kind of hard skills are most needed at the moment for graduates seeking to work in the field, or I guess working in your field? That's where people looking for jobs here. Lots of graduates looking for job opportunities.
I like the industry people go first.
Okay, I could take it, but what I would say is what's most important at any stage is the attitude. The attitude that you should be ready to learn, because what you have learned in the university, when you come into the in a work environment, you should have learning agility to adapt to what's required in a practical area. So just come with a positive mindset. You should prune up your, be clear that learning is important for you to progress. And then listen. When I say listen, listening is important. It may seem very simple, but the initial phase, listening to the customers, listening to the scientists within the company, and to understand how our solutions can address the problems that they are trying to solve. So if these three are the basic areas, I would say, that's required if you come to any company, at least within the life science space.
Makes sense. Thank you, Rajeev. Mark, do you have any comment?
I'll really quickly add, I think what we're going to see in the future is a lot more next generation sequencing for surveillance. I think we're going to see a very large uptick in companies, big pharmaceutical companies, even small pharmaceutical companies in vaccine development, neutralizing antibodies. I think this has forever changed the way that we live. And I think it's clear if you talk to people who run countries that we can't keep having this happen. So we need to very, very quickly be able to generate vaccines, neutralizing antibodies to, you know, to start tackling some of these new and unknown viruses that are going to emerge over time.
Right. So, I mean, I'm interested as well, and I think some of the audience members are also interested in understanding, is Twist Bioscience planning to expand your footprint in Singapore and just, you know, taking a step back away from COVID-19 and SARS-CoV-2 products? What other target markets are you hoping to penetrate in Singapore? Any up and coming technologies that are really, you know, a focus of your company's activities?
Yeah, it's a good question. So we are expanding in Singapore. So we have a headquarters there now. We have a number of employees currently working at our headquarters, but we are expanding. We have open positions, 2 open positions. So if we need anyone out there who's got a protein engineering background, feel free to apply. But yes, we are growing, and we see a number of opportunities in Singapore. You know, synthetic biology, I think, is an area of growth. Definitely taking old manufacturing techniques and re-engineering them using, you know, modern molecular biology techniques within single cell organisms to create, you know, volatile oils, you name it. Anything can really be done. I think imagination is really the only thing that's going to stop us, and we're seeing that around the globe. So I think, you know, Singapore is uniquely positioned there. And I also think with respect to the biopharma industry, we're starting to see a few more biopharmers come into Singapore. And again, I think what's going to happen over time is that vaccine development and, you know, antibody development is going to be something that will fundamentally need to be rejigged and made a lot quicker than it is now, you know, because we just can't afford as a civilization to be able to go through this time and time again. I think, again, it's fundamentally changed the way that we'll approach, you know, pathogens in the future.
Right, thank you. We're still getting quite a couple of questions. So again, I guess for Mark, someone has asked, what are the advantages of hybrid capture as compared to RT-PCR tests?
Yeah.
Bit of a technical question.
Very technical question. So look, I guess first and foremost, you know, what hybrid capture offers over something like a qPCR test is the capability of being able to pick up, you know, new and novel genotypes of SARS-CoV-2 or other pathogens out there that we're targeting. You know, generally, if you're using a qPCR, you design primers that are very specific to a sequence that doesn't generally change. With technologies like hybrid capture, you know, it's more resilient to changes in viruses. It can pull them down, and when it goes onto the sequencer, it can reveal the new sequence. And that allows essentially, you know, to move downstream into something like a qPCR, which is a very rapid and kind of cheaper test. But I think, you know, fundamentally, it allows you to be able to, you know, pick up more genotypes, let's say. And I think the other thing is what we're now starting to see is, you know, rapid multiplexing. Well, some surveillance efforts now are starting to use the large capacities of the MGIs and the Illumina sequences out there, where we can sequence on a single run thousands of individuals. So within a 36-hour kind of turnaround time, you can start screening thousands upon thousands of individuals. And if you need to very rapidly start, you know, doing surveillance, you need that capability and a very quick turnaround. So with something like the respiratory panel that we're coming out with, not only being able to target a specific virus like SARS-CoV-2, but again, seeing what else may be in the population and seeing if there are any co-infections, which would generally help with, you know, a precision medicine type approach.
Great, thank you. Thanks for making it sound so super exciting and something to look forward to as we move forward together as a field. One question for Rajeev. So the question is, has this, you know, whole COVID-19 pandemic changed your sales and marketing plan as a company for the future months coming on in the year 2020 or even beyond that?
Um, I would say definitely, and let me explain why. The traditional structure that most organizations had was you have your direct sales team, and then you have the distribution or distributors as a channel. Then came e-commerce, and these were the three main channels through which we reached our customers. With COVID, we realized that the way forward is going to be a lot digital. And with that comes a great level of opportunity in multiple channels. For example, there are multiple platforms for life science that's available. So if we have to reach out to our customers, we need to leverage those platforms. For distributors, they also today set customers and they sell a set number of products because they are also not that equipped when it comes to training for this entire range of portfolio. So here's an opportunity to build platforms where you can directly connect with customers, understand their workflow, and through that, enable the distributor sales folks to support the customers. Coming to marketing teams, the new way of reaching out to customers would be definitely more and more webinars, as you said. In the past, to organize a seminar, you would get a registration of possibly 100 folks. Recently in Indonesia, we were surprised. We hosted a webinar and we had 600 registrations. Wow. So we realized that the millennials and the youngsters prefer the digital mode, and that's going to be a key focus of our go-to-market going forward. And I think this is what, to the earlier question you asked, for people looking for a job, what do they bring? Their capability in this digital environment, because they are digital natives. That's going to be a big advantage when they come into the organization to help in the transformation.
Right, thank you. I think we can all agree on that. And even coming from the perspective of BCS, we have at first been a bit hesitant with online platforms, and then now we've completely embraced it. Right. So one question for Eong. So somebody asked regarding manufacturing the clinical supply of TY0027, and that manufacturing that happened extremely quickly. And is Tycan already making more of that for phase 2 or 3? And how long do you think that will take?
Yeah, no, we have enough to get into the later phase clinical trials. I mean, obviously, we're not going to, we're very optimistic, but we're not going to go ahead and manufacture everything until we have all the data that shows that it is it's safe and it's going to work. Otherwise, we're just spending millions and at risk, right? So no, that we have enough for what we need to do in the next few months, and then beyond that, we'll just have to let data guide us as to what to do.
Right. Okay. Thank you. Now back at Twist, I think we just I guess it's great that people have different interests and this question for Mark would be, you mentioned something about Twist Bioscience working on data storage, storing movies, files, etc. Could you elaborate more on that?
I can give a little bit of insight, although I'm absolutely not an expert. But yeah, I think what's been seen. I mean, we know that DNA is a great storage device. We find, frozen woolly mammoths after hundreds of thousands of years, DNA still surviving. And I guess that's where the premise of this comes from, right? I mean, if we can take a binary digital platform as we have now and kind of make it, quaternary, let's say, then we can store a lot more in a much smaller space. The biggest problem is really being able to write enough of this quick enough and then being able to read it. So the writing side, that's where we're really working heavily on with a number of different collaborators, mainly out of the US, but there is interest throughout the globe on this. So I, spoke of our platform, currently being able to manufacture 1 million oligos at a time. For this to become a viable kind of option as a storage device, you're talking hundreds of millions need to be manufactured at the one time, and then being able to stitch together properly, which makes the information, you know, readable at the other end. But we think that within 3 years it could be a reality. So we are working on other technologies which allow us to logarithmically increase our oligo manufacture. And we obviously have the technology in the back end to change ones and zeros into ATGs and Cs. So we're getting there, and we think in about 3 years we may have something.
Okay, great. That sounds super exciting. Fantastic. So I think we're about 3 minutes from the end. So I just want to have a last wrap up question for all three of you, which is what do you feel has been the greatest impact that this whole past few months has been on you? It could be a personal, or it could be from the perspective of your company. What has been the greatest impact? What has changed you, I guess?
Look, for me, I generally travel about 70% of my time, and these 4 worlds you see behind me have been my essentially my business place for the past 3 to 4 months. So that's just a quick one for me. But I think what really is shining is the fact that when a real issue like this comes, and it hits globally, it is amazing how quickly companies can come together and start, bringing bright minds together like Prof down here to come out with some novel solutions. So I really hope, I hope beyond hope, that we do come out with vaccines within the next year. But it's not an easy job, but let's hope.
All right, thank you.
For me, I would say it's been fascinating that when a challenge comes, how we come closer, not just as a team, but even with the customers. And we really work to find a solution together. And the second, in fact, there was a friend who was joking and asking me that did the CIO was responsible for digital transformation. I think it's COVID that will be now responsible for digital transformation, the way companies are going to transform. And we for sure, in a big way, would be embracing digital, because that's what our customers want. And we find through this experiment of the last few months, it's been fascinating. It's so quickly everyone got onto the digital wave, and we have been in a seamless way supporting customers and managing our day-to-day work. So these are 2 takeaways for me.
Thank you, Rajeev.
I guess for me, it's just seeing the chance of taking some of the science and into the the translational space from bench all the way to bedside in such a short time. So always something that we've just thinking about, but seeing it coming into actual practice is rewarding. But hopefully it won't take us another pandemic to really iron this process out. Hopefully we'll be able to do this in normal peacetime.
Mm-hmm. Right. Thank you very much. So I'm glad that we kind of ended on a rather positive note. And so I would like to really thank you, Mark, Rajeev, Eong, for taking time to speak with us today, and taking time out of your your activities now, which are really crucial for supporting COVID-19 related research. So I'd like to thank you for being with us. Also to thank Duke-NUS for assisting us, and especially to Twist Bioscience and Merck for supporting this event. I'd like to also thank all the attendees for being here and engaging with us. From the perspective of BCS, we're very happy to wrap up this COVID-19 series. I'd like to share that we will be having a couple of more events over the next couple of months that are focused on workshops. So we'll be having the last regulatory workshop next week, which was part of our regulatory affairs series. That's going to be on the EU medical device regulation. And as of always, if you have any questions or feedback, feel free to share with us. There'll be a survey form that we'll send to you at the end of this webinar. And you can also reach out to us directly. And if you have any sort of burning, outstanding questions for the speakers, feel free to let me or anyone from the BCS team know, and we'll help to direct your questions or interactions. So thank you once again and have a very good evening to everyone.
Thank you.
Thank you. Goodbye.